Thank you for joining the CADASIL Connection speaker series hosted by You've taken a screenshot of the attendee interface, and you should see something in the upper right corner. You were prompted to select your audio and the default your audio in the audio pane, and the dial-in information will be displayed. You've all joined this webinar in listen-only mode, and all callers are automatic. At any time during the presentation, we invite you to type in your question pane of the control panel. We will collect these and address them during the Q&A session at the end of today's presentation. Please indicate if your question is directed to a specific speaker, and it will be read online and addressed by the presenter. Please note that questions are to be in regards to the information presented today that is relevant to the CADASIL community. Individual health questions should be directed to your healthcare practitioner. We will only read 4 questions. Today's presentation is being recorded and will be available on the cureCADASIL website. Again, I'm Jane Gunther, and I'm a volunteer trustee on the board of cureCADASIL Association. In my day job, I'm a scientist working in drug development in San Francisco. My connection to CADASIL is that my partner Bruce and his family have CADASIL. I will start the discussion with a brief introduction to CADASIL and the cureCADASIL Association. Then our first speaker will be Dr. Prenner. She will provide an overview of the known potential disease-modifying therapeutics for CADASIL that are in the early proof-of-concept stage and potential in assessing the effectiveness of potential therapeutics. Dr. Zhao will present her current research in an animal model of CADASIL that supports stem cell factors' importance in brain repair. This is the first webinar in the CADASIL Connection speaker series for researchers and clinicians to present their work in CADASIL. I created this series to encourage communication and collaboration in the CADASIL community at a time where we are seeing years of research from a dedicated few come to fruition. I thank the other cureCADASIL Research Committee members, Sandra Talberg and cureCADASIL President Nancy Maurer, and Deborah Robinson, and our speakers for helping to make this series happen. Our goal is for this to be a unique opportunity for the entire CADASIL community, researchers, clinicians, and families to learn about the state of research in CADASIL, in particular new understanding progression and mechanisms, and the identification of potential therapeutics and biomarkers. Our hope is this series will lead to identification and gaps in knowledge and identify ways to bridge these gaps, to accelerate research and create collaboration that will make a meaningful differences in the lives of those with CADASIL. For those less familiar with CADASIL, CADASIL is a rare genetic disease discovered in 1993. It's caused by mutations of the NOTCH3 gene. It is the most frequent genetic cause of stroke and vascular dementia. Dysfunction of the NOTCH3 protein in the brain vasculature leads to ischemia, infarcts, and demyelination in subcortical areas of the brain, onset of dementia, and physical limitations. Onset of symptoms are generally years, with progression of severity each decade of life. As an autosomal dominant disease of CADASIL in one family member often acts as a shockwave throughout a family, as other affected members come to understand that undiagnosed symptoms are due to CADASIL. The devastation that CADASIL has on families led a group of volunteers to establish the cureCADASIL Association, whose mission encompasses patient advocacy, education, and research support. The dedication and passion of these volunteers to cure CADASIL has led to a number of successes, and some of these are listed on this slide. Our funding activities support research that has resulted in identification of a potential therapeutic and biomarkers by the Dr. Zhao, which was recently published in the Journal of Experimental Medicine in July this year. By establishing a family registry, we transitioned CADASIL research into translation to the clinic. A current initiative is establishing a patient sample biorepository, which will support the biomarker development that is crucial for progressing therapeutic development. Our presenters today will provide more insight on the current status of therapeutic and biomarker development for the treatment of CADASIL. I'm pleased to introduce Dr. Irene Griswold-Brenner. Irene will begin our series with an overview of the known potential therapeutics that are in early proof of concept stage of biomarkers. Irene has made numerous novel contributions in science. Following her graduate work at the University of Chicago and postdoctoral fellowship at UCSF, Irene continued discovery work as a project lead at Elan Pharmaceuticals, where her team identified compounds that prevent neurodegeneration in vitro and animal models for Elan's Alzheimer's, Parkinson's, and multiple sclerosis programs. Irene continued her success in pharmaceutical discovery at Hyperion, where her team utilized iPSC technology to discover the first drug candidate for Alzheimer's disease that was identified using an iPSC approach. This discovery led to a high price tag acquisition of Hyperion by Bristol Myers Squibb, where the drug is currently in phase 2 clinical trials. Following this success, Irene co-founded and is Chief Scientific Officer of Imago Pharmaceuticals, a privately held company that engages in preclinical and clinical development of drug candidates directed against targets implicated in neurodegenerative diseases and fibrosis. Irene has been a productive member of cureCADASIL's Scientific Advisory Board since 2016. I welcome Irene to lead us through the current discoveries of potential therapeutics and biomarkers in CADASIL.
All right, I'll be switching the presentation mode now over to Irene.
This is Irene Griswold-Brenner. Thank you, Jane, for the introduction. And I'd like to say that I've been very pleased to be part of cureCADASIL for the last year. It's an organization I believe will make a true difference in the lives of CADASIL patients. And what I'm going to go over today is just a very high-level overview of what's early stage but potential therapeutics and biomarkers for CADASIL. So there's knowledge is starting to be gained for CADASIL at this point in time, but we still have many gaps in the research and needs to be able to move forward to the point of being able to have clinical trials developed for disease-modifying drug for CADASIL. Currently, there are no treatments for CADASIL. There are no clinical trials that are ongoing for disease-modifying therapeutics, and there are many needs that are in place to be able, things that we need to be able to translate drugs for CADASIL patients. The first is development of a biomarker, or really a sets of biomarkers. And together with this is markers for disease progression or readouts of disease progression. And then obviously we need the drug candidates to progress into CADASIL clinical trials. And previously these have been limited both by funding, lack of organized collaborations, as well as knowledge of CADASIL itself. And we are hoping that this series will help, and cureCADASIL in general will help in this process. So I'll first go over just a brief background of why we need biomarkers for CADASIL. And the first way to describe this is what are the types of biomarkers that one would need to develop drugs for CADASIL. The first is a disease diagnosis, and this is to tell you whether you have CADASIL or not. The second, which is very nice and optimal to have, is a target engagement biomarker. And what this means is it tells you whether you have enough drug in the body and is that amount of drug working as we expected it to. And if you have this in place, then the next thing is to have a surrogate of disease progression biomarker. And what this means is we're trying to figure out if the drug that you're testing is affecting disease. And there are some CADASIL biomarkers, some that have been validated and others that are currently under investigation. The first is the Notch3 mutations. These are a genetic diagnosis of the possibility of CADASIL. The next is to say whether or not the disease has actually manifested or not. And one of these is the GOM, or deposits of granular osmophilic material, which are deposits in your cells that can be detected by histology. The second is MRI imaging that will tell you changes in your brain that are consistent with the diagnosis of CADASIL. These are the imaging and biomarkers that have been are being used in CADASIL. And we also have a number of biomarkers that are under development at this point in time. In Dr. Abuelita Velasquez's lab, there's a couple of potential targeting and engagement biomarkers. The first is insoluble Notch3. We're really excited about this as a potential, as a first step in possibly having something that tells us whether or not the right amount of drug is in the body, and if so, is it engaging its target as it should. And then endostatin may be potentially used as a target engagement biomarker, but also might be as a potential disease progression marker if we see that this progresses, changes over the course of the disease. These biomarkers are still in the animal study stage, as are the rest of these, so they're very early stage. The next one is Notch3. This is being developed in the Oberstein lab. It's a potential disease progression biomarker. It's still in animal studies. And what this is scoring the development of Notch3 deposition in the cells of your body. This is in CADASIL animal studies at this point in time. And a new potential biomarker that's being developed at this point in time is actually in humans, being tested in humans. And this is called neurofilament light chain. In blood, in Dr. Peters' lab, they've shown that neurofilament light chain levels are a potential disease progression biomarker in CADASIL patients, and that the levels of neurofilament light chain in the blood change over the course of the disease in CADASIL patients. This is still a very small study, and we'll have to determine whether this progresses with the disease in a larger number of patients. And then MRI imaging in a number of labs throughout the country and the world, but summarized and reviewed by Dr. Bjornick's lab, shows that the MRI imaging may also be able to be used as a disease progression biomarker in clinical trials. And then finally, Dr. Giraud's lab is now developing functional MRI, which is a way to measure blood flow in the brain and to determine whether or not this might be a potential disease progression biomarker as well. So although we're early stage in the biomarker development in CADASIL, there are a number of potentials that are coming along and that we hope will translate into being able to be either target engagement or disease progression biomarkers in patients, CADASIL patients. And so the next thing I'll go to is discussing what could be potential therapeutics for CADASIL sometime in the future if all progresses properly. But what I'd say first is that because CADASIL manifests with stroke and or vascular dementia, the development of biomarkers or therapeutics for CADASIL, stroke, or vascular dementia each could help the other disease. And what we're hoping for is that if there are therapeutics that are being developed for stroke, dementia, or any neuroprotective therapeutics, that these are some that could be tested in CADASIL and might be able to work on to reduce disease progression in CADASIL patients, or vice versa, that a biomarker or therapeutic developed for CADASIL might work in these related disorders. And the one thing that I would like to make clear is all the studies that are being done to develop therapeutics at this point in time are in animal models, so they are still some ways away from being tested in humans, but we are encouraged that this much development is happening and hopeful that one or more of these will go forward into clinical trials at some point in time for CADASIL patients. So the first study that I'll really at a high level take you through, because we are going to have Dr. Obelita Velasquez give a talk later in this series. His potential therapeutic is a Notch3 agonist antibody. And his concept is, therapeutic concept is that there's not enough signaling through the Notch3 protein in CADASIL patients. And so what he's trying to do is activate Notch3 to circumvent disease by using an antibody that activates Notch3. And what he's found at a very high level at this point in time is that this activating antibody protected a type of cell from death that normally is impaired during disease progression in CADASIL in this animal model. And so we're very excited to see how this progresses, and we will look forward to hearing Dr. Zhao talk at a future webinar. The next one is a similar approach in that, again, it's a Notch3 antibody. This is being developed by Dr. Zhotel's lab in collaboration with Lundbeck. In this case, they're targeting the antibody to the mutated region of Notch3. And their idea is that if this antibody binds to Notch3 in the mutated regions, maybe this will be able to prevent the disease from progressing. And they have tested this antibody in a CADASIL mouse model, and they've reported that it prevents cerebral vascular dysfunction, so cerebral vascular leakage in the CADASIL mouse model. So again, this is another potential therapeutic for CADASIL, similar in the sense that they're both antibodies but different mechanism of action. And then finally we have, before Dr. Zhao's talk, we have one more type of therapeutic that is of interest that we're looking at, that the field is looking into. And this is an exon skipping approach in which the Notch3 cysteines that are mismatched, there's an attempt with the exon skipping to correct this mismatch of the cysteines. And if this translates into CADASIL patients, in theory, this approach could correct the mutations that are present in 94% of CADASIL patients. This study in Dr. Oberseim's lab at Leiden University is very early stage. They've only tested this exon skipping approach in vascular smooth muscle cells, but so far they have shown evidence of exon skipping to correct the mutation in Notch3. So we are looking forward to this being able to progress into animal studies and hopefully eventually into therapeutics for CADASIL patients. And then the next one is the topic of our discussion today. This is with Dr. Zhao's lab, and she will be talking about bone marrow stem cell factors to enhance brain repair. And I won't go into any more detail on this since Dr. Zhao will be talking about it in detail, but I would first like to introduce Dr. Zhao. Dr. Zhao received her MD from Hebei Medical College in China and received a PhD as well at Wallenberg Neuroscience Center at the Lund University. She went on to do postdoctoral work at the University of Minnesota as medical school and has been an assistant professor at Northwestern University in Chicago, as well as an associate professor at Louisiana State University, and is now in a tenure-track position as associate professor at SUNY Upstate Medical University in the Department of Neuroscience. And Dr. Zhao's research has been extensive and very promising in the area of brain plasticity, and using this together with stem cell biology and responses to brain injury, and related specifically to this lecture, stem cell factors, stimulating factors in brain repair, and using both live brain imaging and live cell imaging to analyze the effect of these factors in brain repair. And her research has been important in understanding the pathophysiology both of CADASIL and Alzheimer's disease, and to try to develop therapeutics for these diseases. And we were very excited to hear that Dr. Zhao received the 2017 Bernard Sandberg Memorial Award from the American Society for Neural Therapy Repair for research that has advanced the understanding of how stem cell factors contribute to brain repair, and as well as to her work in stroke, vascular dementia, traumatic brain injury, and Alzheimer's disease. And her research includes the first demonstration of neuroprotective properties of stem cell factors in GCSF in treating both acute and chronic stroke in animal models. So please welcome Dr. Zhao as she presents discoveries from her lab on stem cell factor and brain repair in CADASIL.
All right, and Dr. Zhao, I will be making you presenter now.
Okay. All right, show my screen. Hello?
Hello.
Everybody hear me?
We can hear you.
Okay. Okay. Thank you, Erin, for your very kind introduction. Let us start with why we search for the therapeutic approaches for CADASIL. As mentioned earlier by Jan, that CADASIL is the most common condition of hereditary stroke and vascular dementia in adults. And CADASIL is caused by NATCH3 mutation that lead to progressive degeneration of smooth muscle cells of the small arteries in the body, but this degeneration mainly affect in the brain. CADASIL mainly affect middle-aged adults and cause severe and long-term disability and mortality. The exact process as to how CADASIL develops is still not fully understood. Currently, there is no any specific therapy or treatment that can slow or stop the pathological progression of this disease. The scientific term of bone marrow stem cell factor is called hematopoietic growth factors. Stem cell factor, SCF, and the granulocyte colony stimulating factor, GCSF, are the essential hematopoietic growth factors. Numerous studies have revealed that SCF in combination with GCSF has synergistic effects in maintenance of bone marrow stem cells and in generation of blood cells. Over the past decade, we conduct many in vivo and in vitro studies. Our research findings have demonstrated that SF in combination with GCSF also show a synergistic effect in promoting neuronal network generation and blood vessel regeneration, inhibiting neural loss, and improving functional recovery in animal models of chronic stroke. So then we start to examine the therapeutic efficacy of SF plus GCSF in a mouse model of CADASIL, and to determine whether the combination treatment of SF-GCSF could restrict pathological progression of CADASIL. The animal model, the mouse model of CADASIL used for our study is the transgenic mice carrying a full-length human NATCH3 with origin to cysteine missense mutation at the amino acid position 90. So this CADASIL associated NATCH3 mutation is driven by a smooth muscle cell promoter called SM22 alpha resulted in expression of mutant NATH3 genes in the vascular smooth muscle cells. This model was created and characterized by Dr. Andrew Cosgrove. These transgenic mice develop vascular degeneration and vascular dysfunction in small arteries, include both the tail and the brain arteries, beginning at the age of 10 months. In addition, these transgenic animals also show some pathologic features of CADASIL patients, such as deposition of extracellular domain of NATH3 receptor, GAM, and other extracellular matrix proteins in small vessels of the brain. So to examine the therapeutic effects of SF in in a mouse model of CADASIL, we carry out a long-term study. In this study, to track bone marrow-derived cells, the bone marrow for the mice carry NATH3 mutation was destroyed by irradiation. Then the bone marrow of the mice carrying green fluorescent protein was transplanted to the irradiated NATH3 mutated mice. So this bone marrow transplantation happened at the age of 8 months. As mentioned earlier, in this animal model, they develop vascular degeneration at the age of 10 months old. So therefore, we decided to give the first treatment of SF plus ZF at 1 month before, that means 8 months, at age of 8 months. So SF and GCSF were injected under skin for 5 days, and the same treatment was repeated additional 4 times up to the age of 20 months. In order to evaluate cognitive function, so we use the water maze test that was confirmed 1 month after second treatment and 4 months after the fourth treatment. To label the neuroproliferating progenitor cells or neurostem cells in the brain, BRD injection was provided before sacrifice the animals at the age of 22 months. And then the tissue samples were processed for histological evaluation. This is just a schematic image to show how we set up the water maze test. First of all, we put non-toxic white paint into the water to cover the platform. And also, we put some special signs on the wall of the water tank that allow the animals to navigate where the platform located, and then they can escape from the water. So we use white type mice, which do not express NATH3 mutation as normal mice in our study. So as we can see, during the 5 days testing of the water maze, the white type mice spend shorter and shorter time to find the hidden platform. So that suggests those animals with normal spatial learning and memory, and this is the normal learning memory curve. But however, this learning memory curve never found in the animals express NATH3 mutation at 11 months and 9 months old age. However, SCF plus ZF treated animals found the platform significantly faster at day 4 as compared to the non-treated NAGLU3 mutated animals. The similar findings was observed at 4 months after fourth treatment at the age of 19 months. So those data suggest that ISAF applied as a treatment improves spatial learning and memory in NAGLU3 mutated animals. In the tail artery, so we found the thickness of the tail artery was significantly reduced in the animals carrying a NAGLU3 mutation. However, the reduced thickness of artery wall completely prevented by ISAF applied as a treatment. In the brain arteries, we found the number of degenerative arteries with almost a single layer of smooth muscle cells was significantly increased in the brain of NAGLU3 mutated animals. And this type of degenerated small vessels was significantly reduced by ISAF applied as a treatment. So those data suggest that ISAF applied as a treatment inhibits vascular degeneration, vascular smooth muscle cell degeneration in NAGLU3 mutated mice. As I mentioned earlier, one of the key pathologic features of CADASIL is the deposition of extracellular domain of NAGLU3 receptor in small vessels of the brain. NAGLU3 receptor is a transmembrane glycoprotein. PAS staining is the method to detect the accumulation of glycoproteins. In clinical study, in the brains of CADASIL patients, intense PAS reactive arteries are always accompanied with degenerative vascular smooth muscle cells, suggesting that accumulation of glycoprotein materials in the vascular smooth muscles may play a pathological role in CADASIL. So in our study, the intense PAS positive vessels with abnormal changes were significantly increased in the cortex of the animals carrying NAGLU3 mutation. However, PSA positive vessels were significantly reduced by ISAF treatments. Those data show that ISAF treatment reduced aggregated glycoproteins in brain vessels of the NAGLU3 mutant animals. Recent studies have shown that autophagy can promote cell death. Then we want to know whether the autophagy is also involved in CADASIL or NAGLU3 mutation induced vascular smooth muscle cell degeneration. So use a electron microscope that we found there's many large digestive lymphocytes inside the parasites of the capillary, suggests those parasites are committed to autophagy. To further confirm this is the in vivo data, we isolated vascular smooth muscle cells from the brain of NAGLU3 mutated mice and cultured in cell culture dish. Then we found LC3, which is autophagy protein, significantly increased in the vascular smooth muscle cells of NAGLU3 with NAGLU3 mutation. ISAF plus ISAF treatment significantly, almost completely prevent LC3 production. So those data indicate that ICF plazitide treatment inhibits autophagic commitment in vascular smooth muscle cells with NAGTH3 mutation. So in addition, we also found that the blood vessel density in both the cortex and the striatum were significantly increased by ICF plazitide treatment as compared to the NAGTH3 mutated animals, suggesting the treatment also play a role in increased angiogenesis. Interestingly, we also know that the capillary thrombosis and endothelial cell replacement by bone marrow derived cells in the brain of NAGTH3 mutant mice. In this image, green represents bone marrow derived cells, and the red represents the CD34 positive endothelial cells. If both if the cells co-express both endothelial cells marker and the GIP bone marrow derived cells marker, they turn to yellow. So this is a 3D image showing that yellowish capillary from the cortex of NAGTH3 mutated mice. So as we can see, the yellow stuff and the green stuff completely block the lumen of the capillary. If we cut the capillary at that level, so we found degenerating endothelial cells mixed together with blood cells, and the mixture stuff completely block the lumen of the capillary, suggests that those are capillary thrombosis or capillary blood clot formation. In the downstream of the blood clot formation area, so we found the endothelial cells co-express with GIP. That means that these cells are bone marrow derived endothelial cells. So this suggests that the endothelial cell next to the capillary blood formation may undergoing a degeneration. That's why the damaged endothelial cells were replaced by bone marrow derived cells. So when we quantify those yellowish capillaries in the brain of NAGTH3 mutated animals, we found ICF in the both cortex and the striatum. We found ICF plazitide treatment significantly reduced the number of those yellowish capillaries in the brain. In addition, the length of the yellowish capillaries also significantly reduced by ICF plazitide treatment. So those data suggest that ICF plazitide treatment reduced capillary thrombosis and endothelial damage replacement in the brains of NAGTH3 mutated animals. In addition, we use IgG to visualize the capillary leakage. This is a 3D image showing that IgG were expressed inside the blood clot and also outside the capillary clot. The IgG labeled capillaries were significantly increased in the brain of NAGTH3 mutant animals, and ICF plazitide treatment significantly reduced the number, significantly reduced IgG positive capillaries, suggesting that the treatment may reduce the capillary leakage in the brain of the NAGTH3 mutated mice. Accumulating evidence has shown that there is a tight link between neural stem cell maintenance, neurogenesis, and the structure and the function of the blood vessel system. So in addition to the vascular smooth muscle cell degeneration, capillary thrombosis, and endothelial cell damage, we also found neural stem cells got lost in the brain of the NAGLU3 mutated mice. So this image shows that the BRDU labeled neuroprogenitor cells in the subventricular zone in young and age of white-type animals and in the animals that carry NAGLU3 mutation in treated and non-treated brain. So the data shows an aging-related reduction of BRDU-labeled neuroprogenitor cells in the subventricular zone. In addition, we found the number of BRDU-labeled neuroprogenitor cells in the subventricular zone was significantly reduced in the animals that carry NAGLU3 mutation gene. As CAF applies as a treatment, significantly increased the number of BRDU-labeled neuroprogenitor cells in this area. So this suggests that SF applied treatment reduced the loss of neuroprogenitor cells in the brain of NAGLU3 mutated mice. We then use double counting to visualize the newly formed neurons in the subventricular zone, and we found the double counting labeled cells significantly increased by SF plus SF treatment. So this data indicate that SF plus SF treatment promote neurogenesis in the brain of NAGLU3 mutated animals. In addition, we also found tunnel positive apoptosis were significantly increased in the cortex of the animals with NAGLU3 mutation, and the SF plus SF completed to prevent the tunnel positive cells in the cortex, suggesting the treatment can prevent apoptosis in the brain parenchyma of NAGLU3 mutated mice. We also found that SF TSF plus TSF treatment enhances neural network remodeling in the brains of NAGLU3 mutated mice. We use MIB2 to visualize the dendrites of the cortical neurons. We found MIB2 labeled dendrites were significantly increased by SF plus SF treatment in the frontal cortex. So this suggests that the SF plus treatment play a role in increased neural network regeneration. In addition, we also found the cortical neurons co-expression phosphorylated more ERK. This type of cells co-expression work and the neuron was significantly increased by SSI plus SF treatment in the animals express NAGLU3 mutation. So those data make us to raise a hypothesis that the ERK signaling may be involved in the regulation of SAF plus SF enhanced neural network generation. To test the hypothesis, we carried out an in vitro study. In cortical neurons, we found that the level of phosphorylated ERK significantly increased by SF plus SF treatment. And the SF plus SF increased phosphorylated ERK significantly completely blocked by 2 ERK inhibitors. In addition, we also found that SCF plus CSF treatment increase neural outgrowth and neural network formation, and the SCF plus CSF enhanced neural network formation completely blocked by the two MERT inhibitors. So together, this data suggests that SF plus CSF treatment enhance neural outgrowth and network formation through MERT signaling. So in summary, so our research data suggests that autophagy in vascular smooth muscle cells, the parasites, and capillary endothelial cells damage, and the loss of neural stem cells, progenitor cells, and neurogenesis may be involved in pathogenesis of CADASIL. Our research data also show that ICL plus CSF treatment improves cognitive function, suppresses vascular degeneration and cerebral capillary damage, increase cerebral vascular density, reduce apoptosis in the brain, enhances neural network remodeling, inhibits loss of neuroprogenitor cells, and promote neurogenesis in mouse model of CADASIL. Our research data also suggests ICF plus CSF has therapeutic potential to treat CADASIL. It's worth noting that the combination of SCF plus CSF can be easily translated into clinical trials because this combination treatment has been approved by FDA for cancer patients after high dose of chemotherapy to enhance bone marrow stem cell recovery. In our future study, we will make some efforts on clarification of how CADASIL develops, and we'll further determine how SF plus CSF restrict the pathological progression of CADASIL. We'll keep searching for other therapeutic approaches for CADASIL. We will also validate our lab research findings in tissue samples of CADASIL patients as immediately request. We're looking for these tissue samples such as blood samples and skin biopsy from CADASIL patients. If you have these samples already in your hand, or if your CADASIL patients who waitingly donated these samples for research, for support of research, please contact us. Contact me, actually. So as a long-term goal, so we will move our research forward toward to clinical trials for develop a therapeutic strategy approach for treating CADASIL. This is my contact information. Before I complete my talk, I would thank all contributors for this study in my lab. And I want to thank Dr. Zhao, who provide the original breeding pairs of these transgenic animals carrying a human NACHT3 mutation. This study was supported by American CADASIL Foundation, American Heart Association, and the endowment of Dan Newsom's family. Thank you so much for your attention. Any questions?
All right, Jane, we will be turning the seminar back over to you now, and I will be projecting the next slides from my screen.
Thank you. Thank you, Irene and Dr. Zhao, for your very informative presentations. We are now going to begin answering, I'll be presenting some of the questions I've received from today's presentation. And as a reminder for the audience, you can still submit a question through the questions pane on your attendee control panel.
Okay.
So, question for Dr. Zhao.
Yes.
What would be a potential biomarker for studying the efficacy of these factors in a clinical trial? Perhaps Ring can weigh in as well.
At this point, we don't have anything as a candidate for a biomarker for a study yet. As I showed in my presentation, we evaluate at different levels, in vitro, in vivo, imaging, behavior, and so on. As a CADASIL is very complex disease, and the exactly pathological generation of the disease is still remain unclear. That make it's kind of difficult really develop some treatment. I think in this case we're just lucky based on what we found in chronic stroke study, and then we just want to try whether it works. At this moment, although we have some data generated in our lab, but I think it's so far still far behind to clarify how it works and what we can find, what is the key protein, or whether this is the whole system involved in restrict progress generation or pathological progression of CADASIL in animal models. Be honestly, we don't have a clear answer yet.
Okay, thank you.
Thank you.
I will ask, Debbie, could you put Dr. Zhao's contact information up again? We've had a number of requests to see that information again. And then while that is coming on up, Dr. Michael Wang from the University of Michigan has written a comment and question in, and he says he's impressed with the penetration of the phenotype model, the penetration in your phenotype in your mice model. And I was wondering what background strain your R90C mice are in, and whether you see a strain-dependent effect.
Hello?
Hello, did you hear the question from Dr. Wang?
Oh, yeah, this question for me?
Yes, it is for you, Dr. Zhao.
Oh, I'm sorry. So I thought it's the question for somebody else. I'm sorry. Would you please repeat it again?
Yeah, I can repeat this. Yes, his question is that he's wondering what background strain your R90C mice are in, and whether you see a strain-dependent effect.
Oh, this is animal strain dependent or not?
Do you see a strain-dependent effect?
Yeah, actually, you know, so all the data which I showed today, actually, we use the animal, that's the particular animal model, you know, so actually generated by Dr. Zhao's group. At this moment, I mean, every data depend on this condition. So we're happy to try in other animal model for CADASIL. So if it's available for us. Thank you.
Thank you.
And then we do have Dr. Chow's slides back up. Dr. Chow, if you could advance your slide back to your contact information, please.
Okay. Yeah.
Thank you.
And we have another question here from Dr. Zhao Velasquez. Let's see. What do you think is the next step in therapeutic development of this modality of treatment towards a clinical trial? And I'd like to add on to that question because I work in drug development, and always a big question is how to get enough drug to the target. And the target in this case is the brain. So I was wondering what was the route of administration that would be for a clinical trial.
Okay, thank you. Yeah, thank you for the question. Actually, previous work already showed that both ICF and GCSF can pass through the blood-brain barrier. So then we also, our previous publication also show that ICF and GCSF have a direct effect to modulate neural network formation or neurite outgrowth in cell culture. So we know there's a direct effect for ICF and GCSF to regulate or to manipulate or to reorganize neural networks in the brain.
Okay. And another question, so I'm going to repeat Dr. Zhao's just to make sure that he gets answered. His question is, what do you think is the next step in therapeutic development for this modality of treatment towards a clinical trial? So not necessarily what will be the clinical trial, but what type of steps do you need to be taking in order to be able to proceed towards having a clinical trial?
Okay, so I have contact with the NIH special officer who responds for preclinical trial and phase 1, phase 2 clinical trials. And since our research is a repurpose of ICF or GCSF in brain repair in several disease conditions, today I just present our data to restrict degeneration or pathological progression of CADASIL condition in animal models. To my understanding, number one, we should have both in vivo and in vitro data. We should provide convince evidence to show how it works. Then we can apply for preclinical studies to support our research in this purpose. And then we also need to identify in the preclinical trial stage to test the dose, the effective dose, and also the toxic, the safety of those treatment in animal models. Use an animal model of chronic stroke. Actually, we have tried 6 different doses. We found dose-dependent effect for brain repair and the condition of chronic stroke in animal models. So there's a lot of preclinical data, preclinical studies need to be done before we move forward to clinical trial.
Mm-hmm. So it's good to hear that you do see the dose dependence in the animal models. It's always very encouraging that there's true pharmacology there.
Yes, and so just to keep previous question. This is a model related. This condition limited something. So that's why we seek for another model to validate our finding, and we also want to use CADASIL patient examples to see, to validate or confirm what we found in animal models. So that will increase our understanding whether it works or how it works.
Thank you. It's very exciting.
Thank you. Mhm.
I'm not seeing any more questions from our audience. If you would like to submit a question, you still can submit a question. And Debbie, perhaps you can go to my last slide.
All right.
There we go. There we go. Thank you. So in closing, we hope you can join us at our next webinars that are listed here on this last slide. Our presenters will be Dr. Boehm from NHLBI. He's the principal investigator of the NIH CADASIL disease discovery clinical trial that started this summer. Dr. Ortiz Garcia of the University of Chicago Medical Center, and Dr. Joe Abelita Velasquez of Harvard Medical School and Shepton's Eye Research Institute will be in February 2018. We'll be sending out an announcement email in advance of our October webinar. Our goal is to connect the CADASIL community, so please contact cureCADASIL@gmail.com if you would like more information from our speakers today, or if you have a topic of interest. Once you leave today's webinar, you will receive a survey on the presentation, and we would appreciate if you would complete that and provide your feedback. It's a short survey. I think it's 3 questions. The recording of today's webinar will be available at our website. Thank you to today's speakers for the time they've donated to advance work in CADASIL and to make today's webinar a success. And thank you for your participation today. This is the conclusion of our webinar.
Thank you, John.
Oh. Oh, man. Oh, she must not be there.